the sharing option to Partha.
Yeah, so thanks, Aruna, and I want to echo what Aruna just said. Appreciate all your interest in this topic and your dedication, particularly those who um, completed the assignments, and special hats off to those who had the time, took the time, and made a commitment to to try all the questions and the exercises. So I got a chance to review all of your assignments that you turned in. As a commitment, what I plan to do is over time, I'm going to send to you, and thanks to Aruna, I have at least your email addresses. I'm going to share my thoughts individually with you, so you can use that as a way of learning. It's a continuous learning effort. As I say, Pharm, what we do is not a static science. There is no unique solution necessarily. It's situation dependent, and there are other factors, sometimes internal, sometimes external, that shapes our field and what we do and how we decide what the right choices are. So as you all are convened here today, I thought we'll adopt a slightly different way of giving you feedback on these exercises. And what we have done is selected few of you to address some of the questions. So Ramaya is going to talk about the BABE exercise, and after each exercise, maybe take 10-15 minutes to present what you have. Then open the floor up for Q&A, and I'll also add in my thoughts on what I think are critical factors or things that are missing. But feel free to chime in and share your thoughts. Many of you have done that exercise, so let's make it more interactive. Vinay is going to present the DDI exercise. Again, we'll follow the same format. And finally, I saw that Sujit, Viswas, and Sharmishta Datta are two that I had I reviewed their input on the first in human study design question. I think both of you have approached it from slightly different angle, but if you could share what you did, how you approached it, what your thinking is, and as always, I'll chime in, and others please participate. There are many others, at least 9 individuals I counted who took the time to do all 3 exercises. I would definitely like to hear their thoughts at this meeting. This is, as Aruna said, we are, one effort is to bring you the latest science that we can tap into to improve the quality and expose everybody to what's happening out there in the world. But the other thing is also to build up this pool that you can feel free to reach out and each other to an extent that what each other's strengths and weaknesses are, and leverage that to everyone's benefit. So it's like empowering others to empower yourself is the motto. I kind of paraphrased it a little bit, but that's the focus of this this effort here from Project Dontabhaktuni, and all of us who participate in this process. So thanks all. Maybe I'll invite Ramya to share her screen and walk us through the exercise that she did, and then entertain some questions from the participants today.
So is my screen visible? Can I make sure that?
Yes, it is visible and we can hear you.
Yeah.
Please go ahead.
So good morning, good afternoon, and good evening to all the participants here. Firstly, I need to thank Dr. Aruna and Dr. Partha for giving me this opportunity to present. And I also like to thank Dr. Rajan for encouraging me throughout this process. Now we'll get into the discussion of exercise 2, that is bioavailability and bioequivalence. So going to the background of the drug, this is compound X, which is a small molecule. The previous clinical studies of this compound is carried out in a capsule formulation. And now they have reformulated to a tablet formulation. So there are a few other information about the compound X, that it is a inducer and inhibitor of CYP3A4 enzyme, and it is also metabolized by CYP2C9, 2C19, 1A2. It is a BCS class 2 molecule, and it is having some cardiovascular liability, and it is having a long elimination half-life, and it is also a highly protein-bound drug. So after receiving this question, my mentor is Dr. Aruna, so I discussed with her. So firstly, we went through the guidelines given by the FDA and EMEA. So reviewing these two guidelines will answer most of the questions given in this assignment. And I also went through few sites that will be helpful in answering these questions. And I also went through few similar protocols in clinicaltrials.gov. So firstly, we'll go with the definition of bioavailability and bioequivalence. Bioavailability of a drug is defined as the rate and extent of drug's absorption from the site of administration to the systemic circulation. And absolute bioavailability is comparing the test product with that of an IV as reference, and whereas relative bioavailability is comparing the test product with that of its oral standard as a reference. So relative bioavailability is also called as comparative bioavailability. And two drug products are said to be bioequivalent when, if they are pharmaceutical equivalents or pharmaceutical alternatives, if their bioavailability of the two medicinal products after administration in the same molar doses are similar, and it also shows same efficacy and safety, then the two drug molecules are said to be bioequivalent. So first I'll go with question number 2, and then I'll come back to the first design. So do you need a bioavailability study or a bioequivalence study? So to this question, I'll take you to a scenario in a drug discovery. They will identify the lead molecule. Here, the compound X is formulated as a capsule, and they will do a bioavailability study for the capsule. And if it is showing low bioavailability, then there will be further chemical changes or physicochemical changes, and the drug will be made to a good bioavailability. Only then it will be taken for further developmental process. So in the clinical study, after doing few clinical studies with a capsule formulation, now the team has decided to reformulate it to a tablet. And then the tablet they are planning to approve this tablet and submit it for the NDA, so new drug application. So for in this cases, that is the before the pre-approval process, if there is any changes in the formulation, advice capsule to tablet, FDA recommends bioequivalent study. So we need to see whether the bioavailability of tablet is same as that of the capsule. So to answer to the first part of this question, study. And what are the key differences? So bioavailability of the study, it focuses on one drug, its rate and extent from the site of administration to the systemic circulation. Whereas bioequivalence, it compares two drugs, either if there is a formulation change pre-approval or post-approval before NDA or ANDA submission. And the data of the bioavailability, it provides information about the fraction of drug absorbed as well as its distribution and elimination. Whereas the data of bioequivalence, it compares whether the test product exhibits same rate and extent of absorption as that of a reference product. And bioavailability, it is generally documented after measuring the drug concentration. The systemic exposure profile of a drug is documented. In bioequivalence, the systemic exposure profile of a test drug is compared to that of a reference drug. And lastly, the bioavailability study, you should measure both the parent and metabolite if it is analytically feasible, whereas in bioequivalence study, it is generally not recommended to measure metabolite. Only the parent drug will be sufficient. So these are the key differences between the bioavailability and the bioequivalence study. And the next question is, enumerate few key design features. So there are main design that are used in the bioequivalence study are parallel design, crossover design, and balanced incomplete block design. So parallel design is carried out for drugs which are having long half-life, where the subjects are randomized into two groups, where one group receives the capsule and another group receives tablet. Likewise, it is the pharmacokinetic parameters are compared between these two groups. And crossover design, there the subjects are randomized into blocks, and it is made sure that each blocks and each subjects receives both the formulation with a proper washout period, that is approximately 5 half-lives. And balanced incomplete block design are carried out to compare more than 3 formulation, and it is almost similar to that of crossover design, where here subjects are randomized to blocks, and here it is not necessary that all blocks should receive all 3 formulation. So these are the main key designs that are used in bioequivalence study. And the next part of this question is explain your choice. So in practice, we see all the subjects show large source of variability in plasma or blood drug concentration. So this will be a big bias for the statistical intervention. So considering the importance of this within subject variability, crossover design will be the most appropriate one, where the subject itself acts as his or her own control group. So it will reduce the within subject variability, and we can make the statistical inference without bias. So that is the mostly used design for bioequivalence studies. Now I'm designed the study for this particular exercise. So considering the long half-life of compound X, I decided to choose the parallel study design. But moreover, FDA recommends that even you can carry out crossover study design for drugs with long half-life, particularly in case where you don't have any problems with washout period, subject compliance, or dropout rates or sampling interval. If you don't find any problem in these 3 criteria, you can carry out crossover design for even drugs with long half-life. And it is a single dose study. And then after designing it, the selection of subjects, it should be a healthy subjects. Otherwise, if it is a special population or special therapeutic area where you can select the patients, and it should be 18 years or older. And it also can be based upon the criteria of therapeutic area. If it is for both male and female, you can select the equal proportion of male and female. And if it is like special population like geriatric, you can select that population. And the sample size for the selection of subjects, it's based upon the power calculation, and you can also take into account the error variance from previous pilot study or any previous publications. And sample size also plays major role in the design of the study, whereas if you are selecting a parallel study design, you need more sample. Comparatively for crossover design, you can have less sample. And then preparing the subject. This is a fasted state study, so the subject should be fasted for the whole night, and the drug is administered with 240 ml of water. And then the subjects are allowed to take water all times except 1 hour before and after drug administration, and standard meals are taken 4 hours after drug administration. So after preparing and administering the next point is the sample the subjects. So the blood samples are collected in an appropriate time. FDA recommends for a maximum of 12 to 18 samples. In case in practical impossibilities, you can take into account that more blood samplings around the Cmax area or the sampling interval should cover at least 3 half-lives. So if you can consider these 2 points while selecting the sampling times. But for parallel study design, you should ensure the complete gastrointestinal transit time, and also you need to take account of half-life. So accordingly, it may be approximately 2 to 3 days, and accordingly, you should select the sampling times. After collecting the samples, you need to do analysis, where bioanalytical method validation is another guidelines by FDA, where it talks about the analysis procedures and its validation. After that, we get the concentration and time profile from which we can calculate the pharmacokinetic parameters. The main 2 parameters that are used for bioequivalence study is Cmax and AUC. Specifically for AUC, for long half-life drugs, FDA recommends to use truncated AUC, that is otherwise called as partial AUC, where if the selected subjects having low intra-subject variability, you can use AUC from 0 to 72 hours. It can be fixed to that. But if the subjects are having high intra-subject variability, further discussion with the review group to use the truncated AUC should be done. So I have titled this design as open label parallel study to compare the bioequivalence of an oral tablet of compound X related to an oral capsule after single dose intake in healthy subjects. So this is the profile you get, the concentration time profile after analysis, and you consider this red one as a capsule, that is the previous formulation, and this blue one as tablet. And we need to compare the Cmax, the maximum concentration, plasma drug concentration, and AUC, that is the total area under the plasma drug concentration. So after comparing these pharmacokinetic parameters of capsule and we need a criteria for accepting the bioequivalence. So the criteria is the 90% confidence interval of the geometric mean ratio of the pharmacokinetic parameters like AUC and Cmax. It should be between 80 to 125%. That is, you consider this branded drug as capsule, and if the tablet is showing less than 0.8, it is not equivalent, and if it is more than 1.25, it is not equivalent. Only if the log transformed data and the geometric mean ratio, if it is between 0.8 to 1.5, it is considered as a bioequivalent to that of the capsule. And the last question, what other factors you must consider while designing the study? So the PK parameters of the capsule and how the previous study has been done with the capsule, that information is needed. And the interaction of this compound with that of the food. So the food affects the bioavailability. So if we know the interaction of this compound X with food, we can decide whether we need a fasted study along study. And inter and intra-subject variability in determining the sample size and how we are going to randomize is most important before designing the study. And the availability of the subjects with the duration of the study and the cost of subject and period, how many periods are going to take place, and the dropout rates. So these are the most important things that should be considered while designing the study. And sorry for going back again. I need to point few things in the background. The importance of this inducer and inhibiting activity is not much taken into account in bioequivalence study unless you want to do a combination of bioequivalence and drug interaction study. And the BCS class 2 molecule. So it is considered that class 1 and class 3 molecule, there is another waiver for bioequivalence study, but for class 2 and class 4 molecule, you need to carry out a bioavailability and bioequivalence study. And some cardiovascular liability, in same case, in bioequivalence study, you can monitor the adverse events or you can monitor the heart rate and exposure of the drug. And this long elimination half-life, it is needed for the design of the study. And coming to the high protein bound, so it is considered that free fraction, it is not needed to measure the free fraction of drugs in bioavailability and bioequivalence study unless you give the same amount of dose or you take the same concentration range between the test compound and the reference compound, because you can see here it is just 0.1% of free fraction. So measuring the free fraction of drug will be more unreliable in this case. So you can consider the total plasma drug concentration itself. So that's all my take on this exercise. Thank you for listening. I also request Dr. Partha to make his comment on this exercise answers. Thank you all.
So Ramaya, very well done. Thank you. It's very thorough. I think you did. It shows that you really spent the time to read. Just one thing to add, that we seldom do bioequivalence assessment under fed state, because as you rightly pointed out, food alters not just the absorption part, but it also has other effects on mostly related to gastric motility, the change in pH, etc., etc. So the preferred route is always to do bioequivalence testing under fasted condition. I think it's very, it's very complete. As I said, it's very well thought out, and you have taken into consideration a lot of the factors that the compound property and the background information into your thinking. So I'll open it up to the group here to see if anybody has any questions on this topic.
So if you have questions, you can either put it in the chat box or Arun, can they unmute themselves?
Yes, sir. No, they can unmute and they can ask the questions.
Sure. And while you, if anybody has any questions, please do introduce yourself and then ask your question.
And maybe, Aruna, these slide decks, you can make it available at the website so everyone can access it after the meeting.
Yes, absolutely. So we will actually also wanted to share that today's meeting is being recorded. So we will share the entire meeting on our YouTube channel, and we will also share the slide deck and other details of today's workshop in our Google folders.
Yeah. So, while we are, there are a couple of questions that came up. Let me try to take those on. But what I'll do is we'll capture the questions, and then I'm going to reply to as many as possible offline, and then we can post those responses. So one quick question was, what about the blinding in bioequivalence study? Ideally, we do not do blinding in bioequivalence study because typically everything is done at the same dose level for either the test or the reference product. So we already have a very good idea of the safety. And the reason one does blinding is mostly to mask, interfere with the safety readout in any study or efficacy readout. But since these are very objective measures, you're measuring drug exposure, blinding is often not needed. The other question was that it was stated that the crossover design can be used even when long half-life. Can you please expand on that? So yes, you can do a long half-life drug in a crossover setting. There in the guidance, there is a criteria on how much carryover is allowed. Sometimes under the best of circumstances, you could end up seeing a carryover in some cases if you do a short washout period. And you just have to wait out that. When we talk about long half-life, you have to decide as a kineticist, how long is long enough? Because one of the points that Aruna made is the duration of the study, which is a very critical factor. So you cannot have a study in a crossover setting running for months because that won't be acceptable. So you have to look at the cost benefit and also the feasibility risk assessment when you design these studies. The sample size, you have to look at the intra-individual variability and decide what kind of sample size you need. FDA guidance also talks about how to power these studies, and our statistical colleagues in many companies, one could do that. Even if you look at some of the NCA workbenches, they have bioequivalence module. You can use that as well to see what kind of power you need for the test. Typically, if it is a pivotal study, you want to go in with the highest power as possible. We usually power it between 80 to 90%. But for earlier studies, if it is, if your drug has low variability, then you can get by with smaller sample size. So we will we'll take rest of the questions and answer the questions as they come along. So I think it's time to switch over to Vinay. I have a hard stop at 11, so we have to move this a little bit faster. So Vinay, the floor is yours. I don't know if Vinay is on call.
So yeah, Partha, Vinay is on the call, and I think we have made Vinay the host.
Okay.
So you can start sharing your screen now.
Can you hear me now?
Yes, we can hear you.
Sorry, I somehow missed the email. Good that I got a message and I was able to join. Can you see my slide now?
Yes, we can see and hear you.
Okay, great. Thanks.
You can move it to slideshow mode. Yeah, thank you.
Good evening and good morning, depending on the place where you are. My name is Vinay, and I will take you through the second question that we have assignment, that is the drug-drug interaction study. Background information which is there, I think it is very similar to that is common for all three assignments. First of all, I would like to say that Ramya has given a very good presentation. So this presentation may not be so detailed, but I will try to cover all the key points. So questions that we have in this topic is design CP study or studies that addresses the DDA liability of the compound. Do you have all the necessary information, and what other information would you rather have? So I will cover all these points in my next few slides. So this is the overall objective. Design a CP study or studies that addresses the DDI liability of the compound. So what are the DDI liabilities? When we say DDI liabilities, two questions. One is, can my compound of interest, that is compound X here, can it affect the PK of other concomitantly administered drugs? That's one question, one question, important question. Second one is, can the other compound affect the PK of compound X? This is the other question that we have. So these DDI interactions mainly happens because of mainly two reasons. One is interaction at the enzyme level or interaction at the transporter level. So first, let us go through the first point that we have, information that we have about this compound is, in vitro studies suggest that the compound X is both an inducer and an inhibitor of CYP3A4 at pharmacologically relevant doses. So this suggests that our compound X is an inhibitor of CYP3A enzymes, right? Then what is the impact? It can increase the plasma concentrations of CYP3A substrates if that compound is also concomitantly administered with compound X. So therefore, to assess this, we may have to do a single dose DDI study with a CYP3A substrate. Second point is our compound is an inducer of CYP3A enzymes also. So now what is an inducer? Basically here the synthesis of the particular enzyme will increase. So first the synthesis has to happen and it has to reach a steady state. So what happens if our compound induces CYP3A enzymes? It can actually it will increase the metabolism of the other substrate. So what can happen is it can actually decrease the plasma levels of other CYP3A compounds that we administer. So in order to increase the synthesis of CYP3A and to maintain certain levels, we may have to do a multiple dose study. That is, we'll have to dose multiple times, maybe up to 2 weeks or so, the inducer, so that CYP3A level increases and reaches the steady state. So that's a multiple dose study, and then administer my CYP3A substrate. Other information that we have. So extent of inhibition and induction are dose dependent. So here we can select the highest clinical dose of compound X in our study. Nothing is known about the interaction of the compound X on drug transporters. So it suggests that we don't have to worry about interactions because of transporter inhibition or induction. So we don't have to worry as of now. So compound X is a BCS class 2 molecule. It suggests that it's a low solubility and a high permeability compound. This is just an information in this case because we are going to use a marketed product or the highest dose. This is mainly an information here. Then molecule is also metabolized by CYP2C9, 2C19, 1A2, of which 1A2 is a major pathway, but all three pathways represent 20% of total elimination. So let us go a little deeper here. So overall, it is telling us 20% of total elimination. So basically, overall CYP contribution is on the lower side. So remaining 80%, how it is getting out of the body? By other modes of elimination. It can be through biliary route, or it can be through renal route, or it can be through phase 2 enzymes. So there are multiple other options. So then out of these three, at least 1A2 is a major pathway, it says. So out of 20% is a major pathway. So it may be assumed that it can be say about 10% or so, because we don't have the exact values. So 1A2 becomes important. So but 1A2 is also a polymorphic and also a saturable type of enzyme. So we may have to know in normal metabolizers what happens, or in extensive metabolizers how my compound behaves. So this also we may have to study. That also comes under DDI liability. So and it is also gets induced by smokers. If the people are smoking, that smoking also leads to induction of this 1A2. So that also information will help us. Either we can do a separate study, but that may not be required initially. In the same study, whatever we are doing, there we can identify that whether who are smokers, who are not smokers. And once the study is done, we can do some post-hoc analysis where we can compare whether is there a significant difference in PK between smokers and non-smokers. Molecule has some cardiovascular liability, increase in heart rate and the other issues. So in the study, it suggests that we need to closely monitor heart rate and QTC along with other parameters like blood pressure and other vitals. So what happens here? We are thinking about administering the compound at multiple doses also, right, in the induction study. So as the time increases, like day 1 AUC, and then after day 7 AUC, if you take, day 7 AUC might be much higher than day 1 AUC because of the accumulation and other reasons which we don't know, but it's better to monitor these the QTC and other heart rate parameters. Molecule has a long elimination half-life. We don't know the exact number. I have assumed that it is about 10 hours or 12 hours, 14 hours in that range, but it can be much more than 20 hours also. Of course, this will have a significant impact on the design that you select. That we will come back to that. But for our purpose, I have just considered 10 or 12 hours. Molecule is highly protein bound, 0.1% free fraction. It means it suggested probably 90% bound to plasma proteins. So guidance which helps us to design the study is if from FDA guidance this is the one, clinical drug interaction studies, cytochrome P450, enzyme and transporter mediated drug interactions. So this is a very nice guidance, very in detail they have explained it. So coming to the design, how do we design this study? So first thing is that we can do this study in a healthy subjects. Next question is we'll have to select an index substrate. For index substrate means the substrate which is mentioned in the guidance for CYP3A4, CYP3A. So if you look at this table which is in the bottom of the slide from FDA website, it gives this these are all the index substrates for different CYPs. The last one is CYP3A, and you can see that one is midazolam, triazolam. These substrates can be used if you want to assess CYP3A. So generally midazolam is used because it has got less side effects, and it's a clean substrate for CYP3A2. Around 2 mg is the dose that is used. Number of subjects we can assume as 12 plus 3. 3 is standbys because when you administer it for when the study goes, some people we might drop out also. To support that, we'll have about 3. 12 subjects is on the minimum side, so depending on the statistical and other parameters, you can increase also because of the variability and other data that you have. But it's on the lower side, a good number. Then I'm assuming that compound X and substrate both we are administering once daily, and substrate sampling over 24 hours because midazolam is a very short half-life compound, so that's good enough 24 hours. And inhibitor, that's our compound X, sampling over 48 hours. So because 10 hours half-life I'm assuming, so 4 to 5 half-lives you need to cover. So it comes to about 48 hours to cover 80% of AUC. Then once if you collect all the samples and measure parent levels, you'll get the PK parameters of both the substrate and the parent, substrate and the inhibitor or the inducer, that is compound X. The change in Cmax and AUC and some statistics can be applied. In brief, this can be one of the design. So you can do either parallel or crossover, but for if the half-life is around 10 hours, then we can do a crossover study also. So it's a 3-period, 1 sequence crossover study in healthy subjects. So it will not be it's not a crossover. It's not a what type of it is. Basically, it's 1 sequence, and it's not a blinded study. Right, so basically it has got 3 periods. The first arrow mark which you see here, that is period 1, substrate. So here we can dose midazolam and we can collect the PK parameter, then give a washout. It's a short half-life compound, but still you can give a say 1 week washout. And then in period 2, you can administer the inhibitor first and then the substrate. So here we are doing this study. This phase will tell us inhibition. What happens if my compound inhibits the metabolism CYP3A? What happens to the PK of midazolam when CYP3A4 is inhibited? So that information we get in period 2. We give some short period, and then we need to do an induction study. For induction, we will have to give the same drug that is compound X for about 2 weeks, right? So you give it for about 2 weeks, and then administer the inducer, and then the substrate. Say on the day 14 or so. Day 14, you administer inducer plus substrate, right? In this way, you can do this study. And if you assume that it is a very long half-life compound, like more than 24 hours or so, then you may have to do a parallel design, where in one group of subjects you do only the midazolam. In the other group of subjects, say another 12 subjects, you do inhibitor plus substrate. And then you will have to do the third part of the study in the third group of subjects. Then after the period 3, if you want, you can add one more period you can conduct. That is to understand, like maybe after 1 more week, you can administer only midazolam. So that will tell you whether what happens to the enzyme levels and how many days does it take for the enzyme levels to normalize. So it will tell us the enzyme's activity, how many days it takes to become normal. There is one more point on the bottom of the slide that's also probably very useful. See, here we are administering this compound for so many days. So one thing is what happens to the midazolam levels. At the same time, we can use some biomarkers also. These are basically the endogenous substances. One can include biomarker like example for CYP3A, 4-beta hydroxy cholesterol. So because that's an endogenous substrate, we can collect the plasma samples, and the same plasma samples we can measure. So we will have very good data what happens in period 1, and then what happens in the example in the period 3, day 1 what happened, say first week what happened, second week what happened. So that will also give a lot of good information to understand more about compound X. Yeah, that's all I have. I think any questions are there, please let me know. Thank you to Dr. Partha and Dr. Aruna for their useful comments whenever required.
In the interest of time, I would suggest that we actually move on to the next assignment that Partha is going to cover for us. And if you have questions, please type your questions in the chat box, and we will address your questions offline. In fact, we will actually share the answers for these questions with all of the participants. So, Arun, if you can please make Partha the host again.
Yeah, so what I'll do is I'm going to, Vinay, first of all, thank you. You've spent a lot of time on this particular example, and I think you have captured, the essence of, what kind of study design one should consider in these cases. It's not a simple, straightforward case, and thank you for taking the time to walk everyone through that example. So I don't know if Sujit is on call. What I would like to do is I can put up his response, and maybe also ask others who did the first in human example to chime in. I'm going to also share my thoughts as we go along in this case. So just bear with me 1 second. So can everyone see my screen?
Yes, Partha.
Okay, so maybe Sujit, if you are on call, do you mind taking us through this example?
Hi, can you hear me?
Yes.
Okay, thank you. Thank you for this opportunity. I'm really enjoying this whole workshop. And sorry, I didn't prepare any slide because I didn't know that I have to talk. So briefly, I'm going through the third assignment, like first in human dose. So based on knowledge, actually, I prepared it, and I also went through the FDA website to take some insight from them. So for selecting the first in human dose, the main concern is the safety. efficacy at that stage may not be the main issue to consider. The main issue to consider is the safety, whether the dose is safe for the healthy volunteers or like even for the patient population if it is cancer drug. Because if it is cancer drug, then the target population is always the cancer population. So in that case, also the safety is the first concern. Is there any cytokine storm or some other thing is happening or not? So few approaches are usually adapted to select first-in-human dose. So the most common is like a NOEL dose. NOEL means like non-observed adverse effect in most sensitive animal study, and they used to select that dose for a healthy population based on some elementary calculation. So then another approach is highest non-severity toxic dose in animal, especially for oncology drug. In common, it is called like maximum tolerated dose. So another approach is 1/10 of the severely toxic dose, which is observed in the animal, and that can be used for the first as human dose. And another approach also is at minimally anticipated biological effect level. This is commonly used for most of the cases biological products like macromolecules, but I think nowadays is also applicable for a small molecule too. And another last not the least, like pharmacology active dose. Those doses actually selected based on some PBPK mechanistic study as well as other. They used to use like other tools to find out a safe dose, and that also has like pharmacological activity. So they use in vitro and animal study data to select that dose. So those are the approaches they use. But for my opinion, I want to choose like the last two one, like MABEL and PAD dose, like pharmacological active dose. The reason is because those dose you can get some efficacy information also beside your safety information. So that means you can get like more advanced insight than considering only safety in that sense. So that two approaches, I think, I think more appropriate for designing a study first in human dose that can help you to move forward quickly for the next step.
Yeah. So Sujit, if I may, cut in quickly, I think given some of the recent development in first-in-human studies, the agencies, particularly in Europe, they are emphasizing more on MABEL and PAD, particularly if one knows the mechanism of action. Then they insist that why do you need to go to a maximally tolerated dose and not use the saturation of the biomarker or target engagement markers that you have identified as primary MOA, to drive your dose escalation and maximum dose in your study. So it's getting more traction lately.
Thank you for that. So,
Please continue.
So should I go ahead?
Yes,
Okay.
please.
So okay, so then now I actually completed the approaches which are usually utilized to select the first in human dose. But there's some background information always we needed to reach that step to select the dose for the human. So in my opinion, so the pharmacology of the drug is really essential to select any kind of decision regarding dose. So the mechanism of action, definitely we need to know the off-target effect, the drug-drug interaction, and also like overall PK, like ADME properties. Protein binding also another important aspect, because sometimes, if it is highly protein bound, then over time with the other kind of similar drug, like say for example, the patient is taking another drug which has also similar kind of protein binding, then it can cause it can alter the PK profile, especially like blood concentration based on protein binding. It will be a competition between them. So there's also those information also very needed to get this stage. So based on well-conducted non-clinical PKPD study, especially the toxicity study results, so the MABEL and PAD can be a best choice to consider first in human dose, because I mentioned earlier, those approaches can give you some efficacy information as well with the safety. So the similar thing, actually, those information are missing in that background information. So I mentioned that like we need to know those kind of information to reach that stage. In question 3, that's I mentioned like what are information we might need to approach this stage. So as an answer of question 4. So all the in vitro and non-clinical pharmacology and safety information were missing. So based on this stage, we actually were not able to design the successful first in human dose clinical study, because we need actually those information because we are concerned about the safety. Then for the answering question 5, so what would be the biometrics we can use for sampling or like evaluate the drug safety? So blood and plasma, feces and urine, those are kind of easily available. We can consider those first to measure some safety information. For example, blood, we can do some like hematological biomarker study for the safety analysis. We can do some like urine analysis for the kidney function, feces analysis for the like clearance, especially like hepatic clearance. So those are really important to measure during this first in human dose study, because we can, based on this parameter, we can scale up or scale down the dose in further study. So for the answering question number 6, so in my opinion, I'll choose the staggered dosing instead of cohort dosing, as I don't have the complete risk assessment result to proceed confidently in this scenario. So that is all I did for answering these questions. I'll be happy if you have any insight or any comments or questions regarding this.
Yeah, Shujit, this is Partha. Thank you for taking the time and reading through the guidance and made a great effort in answering these questions. Just one thing about the point question 6 is that under the current guidance that is in and also being adopted by FDA and other countries around the world for first in human, that they are suggesting a sentinel cohort at every dose level, where you dose one subject and with active and one with placebo to test, the safety. And once the safety is cleared, then you expose the rest of the subjects in that dose group. In some cases, we have to do online PK so that the assessment of safety data are done in conjunction with the exposure data. But if we have enough safety margin, then you could allow the exposure data or the PK analysis to stagger a little bit, so that you are not beholden to the bioanalysis timeline, etc. So these are all decisions that are made based on not just the science, but also the operation aspects. So I'll take a pause there. I know there are others, at least 8 others who attempted this question. So could any of you share your thoughts and your questions with this group here?
So Parth, this is Aruna here. So one of the aspects I think that I would like to highlight here is that when you're looking at unmet medical needs, in case of oncology, then we're looking at this first in human study being done in patients rather than healthy volunteers. So in which case, the first human dose that we choose also has to take the patient compliance and efficacy into account. So that is something that as a clinical pharmacologist, we do have a very important role to play here, that you would like to choose a dose that is not only safe, but also a dose that is advantageous to the patient.
Yes, sir. So there, I think many others use Mabel or a little, variant of Mabel. There are some publications now out there. PAD is particularly done in that case because you could choose your starting dose based on what you think is the target engagement or biomarker change you need to see minimum effect in that population. So you could go with EC50, IC50, or 75. Whatever you choose is reasonable. And also, in that case, we take into account the therapeutic index, and that is kind of gleaned from the safety, preclinical safety knowledge, like the exposure at where we start to see, dose-limiting toxicities, and we try to go a little lower so as to give us the room to dose escalate enough so we can reach not only a maximally tolerated dose, but also where we can achieve significant efficacy. So it's that's another thing, and good point that I left out the safety information completely to see whether that's a key feature of any first in human study design, to know where the safety is, what the NOEL is, in what species. Do we see only in rodents, or do we also see in non-rodents? What is that differential in exposure where we see NOEL, where we achieve NOEL, or in rodents versus non-rodents? Species sensitivity, it gets more complex from there on. Sometimes toxicity could be species specific. So, then you probably have to do another higher organism like non-human primates, although it's seldom done. But, there are chances where, instances where you just have to do those studies. And even in some exotic species sometimes, because you don't have that, whatever biomarker you chose as the target engagement marker, it's not, expressed in the species of your, choice, usually the dogs, etc. So, there are one of exceptions for every case that you can imagine in drug development. All right, so what we can do is, please share your questions. We'll collect those and I'll answer, each of those questions, share it with the committee, and, Aruna and others can post it on the website for you to review. And if you have any further thoughts or questions, feel free to reach out to me. I'll be happy to, get back to you with my thoughts. Okay, Aruna, I hand over the floor to you.
Thank you, Partha. And thank you to all our participants who actually took on doing these assignments. I know they were not easy. At the same time, we wanted you to take the time to go review the guidances and familiarize yourself, because as you're moving into taking on roles in pharma companies or wherever you work in the near future, you will have to take on doing this on a regular basis, reviewing the guidances, like really looking at where else should we be looking at to find the answers to the questions. Again, this was a small teaser, so you are well prepared and can take on the roles of clinical pharmacologists in your future careers. Again, briefly wanted to touch upon that we are going to be having another workshop in next month. And this will be, I think there were a few questions that came up with regards to how can we do plotting, what can we, so the focus of this workshop is getting you familiarizing yourself with using R for pharmacometrics, whether it is for graphing or any of the other. And this is just a small teaser to get you started in using pharmacometric tools in your day-to-day science. Again, a workshop of this size would not be possible without the support and contribution from multiple people. This was our attempt to have the mentor-mentee relationships going. And I want to take the time to acknowledge all our mentors who took on reaching out to their mentees. It may or may not have worked out the way you would be perfect, but again, this is all a learning curve for us as well. Thank you all who have chosen to be mentors and who supported their team and were available to answer questions from their mentees. I also want to acknowledge Partha for taking his time, putting in this workshop, and really, I know you had a hard stop, Partha. Thank you for staying with us. I know we're 15 minutes over our time right now. Thank you so much for all your contribution. I acknowledge Arun, who is the primary person who's organizing these WebExes, our co-host, SSX board, and Tanga, who has been instrumental in making these workshops possible. Ramya, Krishna, Ramesh, who have been the primary people communicating with you, and Ramya, who also spent a lot of time in working on those assignments, optimizing that slide deck, and Rajan, who has been the wind beneath my wings to make all of this possible, and who also will be presenting our next workshop. And of course, I do want to thank our sponsors. That is PharmaPro Consulting, which is a consulting company specializing in PK, PKPD, pharmacometrics modeling, as well as regulatory affairs. Without their support, this would not have been possible either. And please take on subscribing to us. Do reach out to us and do continue to participate in our, I apologize that there is a typo in terms of the dates here, but we would love to see you in our future workshops. And do take your time to give us your feedback. You will receive a survey feedback from us. Do take the time to give us your feedback. It's really important because as we are hearing from you, we will work on optimizing our workshops. And our objective is that we provide these workshops to you so you can develop in your career and have, have everything that you're looking for in your career growth and knowledge. Thank you again. Thank you all for being here, and we look forward to seeing you in our future workshops.